Good afternoon, everybody. Today, we were talking with Bob Wildfong from Seeds of Diversity. Bob has been saving seeds for over 30 years and is the Executive Director of Seeds of Diversity, a national organization for seed savers who are rescuing the diversity and heritage of Canada's vegetables, fruit, grains, flowers, and herbs. He is also the horticultural specialist at the Dune Heritage Village in Kitchener, a 40-acre living history site that recreates life at the turn of the century. Thank you very much, Bob, for joining us today to talk about intermediate to advanced seed saving techniques.
Thanks very much. I have a presentation that I'll share. Just give me a sec. And there we go. So thank you very much for inviting me here today. I work with a not-for-profit organization called Seeds of Diversity, which is a seed savers group. We're people all across Canada who save seeds, and the reason that we do that is to rescue the rare varieties that other people aren't growing so that those varieties can continue to exist. This talk here is it's about seed saving. I'll go into all the topics that a seed saver or even a commercial seed grower would have to know about. And it's called Beyond the Basics because I will talk about the basic seed saving, but I'm assuming that people have a grasp of the basics part of seed saving, and we will take a bit of a deeper dive into some of the details of it. First of all, I'd like to talk a little bit about the reason why we save seeds. Seeds of Diversity's interests are as wide as our membership. We have a lot of seed savers, and sometimes people save seeds because they want to save a little bit of money. Sometimes you save seeds because you have a favorite kind of seed that you maybe inherited from a relative, and it's a family heirloom, and you just can't get it anyplace else, so you have to grow your own. I have friends who work at historic sites, and I also work at a historic site in Kitchener. Some of the historic varieties that populate those recreations of old gardens are varieties that you can't buy from any seed catalog. And so we save seeds because that's the only way to get those seeds. Overall, if you look at this circle on the screen here, imagine that represents all the varieties of seeds that you could get 100 years ago. It turns out that back then, we had a lot more seed savers. People saved their own seeds often. And over the last 100 years, we've kind of lost that. We don't save our own seeds as much as we used to. Imagine that you would have your own seeds, you would know how to plant them, you'd know how to grow the plants, you would harvest the produce, and you'd also keep seeds from that harvest and plant them the next year. It was simply just part of what you knew how to do. It was part of the gardening cycle. And for some reason, over the last 100 years, we've kind of stopped doing that one part of the cycle and instead gotten to buy all our seeds in the spring and not save them in the fall, but gone back to buy more the next spring. Since many people were saving seeds, many varieties could be perpetuated just by people saving lots of different varieties. With fewer people saving seeds now, they can save only fewer varieties of seeds. And the consequence is over 100 years, 3/4 of all those seed varieties that existed back then are extinct. We look in seed catalogs, we can't find them. We look in seed banks and we can't find them. They're gone. Out of the 1/4 that still do exist, see the little green triangle? That's what you can buy. We look in modern seed catalogs and compare it to all the varieties that have ever existed, and we can only find, out of even what's left, only about 1/8 is in seed catalogs. So what about the other 7/8? Where are those seeds? They're still alive, but they're in seed banks locked away where you can't get them. They're in people's collections. They're growing them, but they're not available to you. Fortunately, seed savers are growing those 7/8 of varieties in their backyards and keeping them alive. And through seed exchanges and through seed libraries, we're starting to get some of those varieties out there and more available where you can get them. Some of them are even being picked up by seed catalogs or seed companies, and that just makes them easier for people to grow. That's what Seeds of Diversity is all about. We want to get those 7/8 of the seed varieties that we still have, but you can't get. We want to get them out there so that you can grow them. And that means we need more seed savers. And that means I'm here to talk about how to save seeds, and hoping that some of you will take on the challenge of growing a rare variety, save its seeds, and spread those seeds around. Get other people to grow them too. That's the only way we'll save that plant diversity. So in order to save our seeds, there are some things that we want to make sure we do well. We want our seeds to germinate. If the seeds are to germinate, then typically we harvest the seeds when they're fully ripe on the plant. You can't save seeds before they're ripe and expect them to grow. They have to fully ripen when you harvest them from the plant, and then when you plant them, they'll germinate. You have to clean and dry the seeds properly. We'll talk about how to do that. You have to store the seeds properly. Every seed is a living thing. It's a little baby plant inside a shell, just like a bird in an egg, and it has to have some food. So every seed has a little bit of food stored up, and it's a little baby plant eating that food. When the food runs out, the seed dies. That's why old seeds don't grow. So we'll talk about how to store your seeds so that they hibernate and eat their food slower. That's why they'll last longer. We also want our seeds to grow into the plants that you expect them to grow into. You don't want to harvest seeds thinking that you'll have, say, a pumpkin, and when you plant them, you actually get some cross between a pumpkin and some other squash that you didn't expect. So we'll talk about how to prevent cross-pollination between varieties. And we'll talk about a few advanced topics called roguing, which means taking out the off-types in your plants so that you're only saving seeds from the plants that have the characteristics you want. And we'll talk about the population size, which is something that many seed savers don't know about, but it's really important to have an adequate number of plants that you're saving seeds so that you get all of the genetic traits that are within a variety. And we'll explain that. So first of all, harvesting when the seeds are ripe. We find that 95% of the time, seeds are ripe on the plant when they're dry and brown. So think of how beans look when they're ripe on the plant. If the bean seeds are truly ripe, then the pods are brown and dry to the touch, and the seeds inside are hard and dry too. We also find that with all flowers, pretty much all herbs, they're all sort of a brown, flaky kind of handful of seeds and chaff. But the point is, you wait until they're brown and dry. If you were to take flower seeds from almost all kinds of flowers, when they're still green and kind of soft to the touch, they would not grow. Those seeds would just shrivel up because they haven't matured yet, and they haven't gathered enough food, so that the little baby plant can live. Instead, the seed would probably just shrivel up and die within a week or a month. And that's not long enough to plant it next year. You have to wait for the seeds to be fully brown and dry on the plant, because that mother plant is feeding the seed, giving it lots of food so that it can live through the winter. At the bottom, where the picture of the watermelon is, about 5% of our seeds come from fruit that are ripe and moist. Maybe a pepper is a good example. When you slice open a bell pepper, you find seeds inside, and they're moist. Watermelon, obviously the seeds are moist when you harvest them. Tomatoes, other kinds of berries and fruit. But you might think, and I always did, that makes up most of the seeds that you might harvest. In fact, if you count up the number of species, you'll run out of fleshy, soft fruit a long time before you run out of species of flowers or legumes or grains, which are all dry. When we process the dry kinds of seeds, say beans are a good example. There's a big wire mesh at the bottom of this pile of screens. That's perfect for beans. If I crunch up the bean pods and let the beans fall through the mesh and leave the pods on the top, that's a nice way to clean those bean seeds out. Say I'm cleaning lettuce seed. One of these screens is going to be perfect for that because lettuce has little bits of, let me go back, the hand right there that has lettuce seeds in it. So that's a combination of seeds, little bits of flower petals, little bits of stems, little bits of all sorts of things that were near the seeds. The small mesh will allow the powdery bits to go through, but not the seeds. And slightly larger mesh will let the seeds go through, but not the stems. And so in between, we wind up cleaning up the big stuff, cleaning up the little stuff, and if we get the sizes of mesh right, we can catch the seeds in the middle. So these kitchen strainers and deep fryers and pieces of actual fencing that I've made into screens, these are your best friends when you're trying to clean the seeds of 95% of all those seeds that you'll harvest. When we do the same thing for the fleshy fruit, this is called wet processing. Here's a tomato I've sliced open, and the basic and easy way for cleaning off tomato seeds, you see I get that pulp on my fingers on the left-hand side. What I've done is I rubbed that pulp with just a cloth, just a towel, and wiped all the jelly off of those seeds. You can imagine when you try to pick up a tomato seed from a cutting board, how hard it is to pick them up. They're covered with this slick jelly, which is actually a germination inhibitor. A few times, just to see what would happen, I've allowed the seeds on the left with that jelly on them to dry. What happens is you get a kind of a red fruit leather with seeds embedded in it, and that seems okay. I thought, well, I could just plant it then. And in fact, it repels water when it's dry. It takes a long time for those seeds to grow. And the germination rate was really quite low with that jelly on it. Its purpose in nature is actually to prevent the seeds from germinating right away. When a wild tomato drops a fruit on the ground, it doesn't want that. It doesn't want the seeds to grow right away. It wants them to wait for a little while because some cold weather will come. But in the tropics, where tomatoes come from, it's not cold enough really to freeze. It's just cold enough to kind of slow down the rotting and fermentation of that jelly. Here, we can't have tomato seeds outside too much in the winter because they are tropical and they tend to freeze to death. But when we take our seeds indoors, they don't have the opportunity to self-clean. We have to clean them off ourselves. So the tomato seeds over on the right that are nice and clean, they'll get 100% germination right away. It's just a matter of cleaning them off. Now, what if I have lots and lots of seeds? What if I am saving thousands of tomato seeds, which a seed company would do and a seed bank would do, and Seeds of Diversity's seed collection, we do that. We collect a lot of seeds every year. I don't want to rub them all with a cloth. Instead, I let nature do this. So here I am squeezing the pulp out of a tomato. And I'm smart enough that I squeeze the pulp with the seeds into one container and then put the rest of the tomato in another container, and that goes into nice bags, which I freeze for eating. So that goes into my freezer, but the seeds sit in this container. I put a lid on it and let it sit at room temperature for 3 days. Now, 3 days is enough for this to happen. The pulp has gone moldy. It smells terrible. What's happened is that the jelly all around the seeds has rotted away, and the seeds have sunk down to the bottom, all nice and clean. So I can put a little bit of water in here and gently pour off that stinky pulp. And guess what? There are the seeds. They're nice and clean, sunk down to the bottom, and the pulp just kind of pours off and washes away. Another rinse, and look how clean they come. That's way easier than rubbing them with a towel. And then I put them in a strainer. You can squeeze the seeds a little bit to get the water out, and then I let them dry. This is exactly how a seed company would wet process a large amount of tomato seeds, thousands and thousands of seeds. They might not use a yogurt container. They might use something like a garbage can instead. And they might not use a little kitchen strainer to strain them out. They might use a great big screen instead. But it's essentially the same technique and the same technology, just a bigger scale. So what we're able to do in the home is really exactly what is done in a seed company. It's just a matter of adapting the scale. But the quality of these seeds will be exactly the same as if you'd bought them from a seed company, because they were processed in exactly the same way. Two things that are really important in these pictures. I'm using a bowl, just a nice smooth kitchen bowl, not paper towel, which sometimes people say you should dry these seeds on paper towel. What tends to happen is that the tomato seeds stick to the paper towel, and you'll spend the next few days trying to pick all the seeds off of the paper towel. I like to use a nice smooth surface. A baking tray works really well. Wax paper works really well. Bowls are nice because as the seeds dry, I don't want them to kind of fly away off the edge. So the edges of the bowl hold the seeds where I want to keep them. And the second crucial thing is the label. These happen to be a variety called Watermouth. And that is a crucial label because I can tell one kind of bean from another if they're colored differently. And I can sometimes tell the difference between different kinds of seeds that have different sizes. Maybe a tomato with large seeds and a tomato with small seeds. But that's getting to be kind of tough even for me. I can't tell the difference between different kinds of tomato seeds. And I don't have a good enough memory to remember which one is in that bowl. So always label everything. Now, what if we find that not all the seeds in that tomato were actually fully pollinated? If a seed isn't actually pollinated, then it's kind of going to be poorly formed. You'll find that it's a little smaller than the others, and it won't grow. Imagine we have 5 seeds, and one of them just happened to not be pollinated properly, and it won't grow. That means if we planted those 5 seeds, we'd get 80% germination. And here's a trick. This is what seed companies do. They figure out how to get rid of that one, because then their seeds will be 100% germination. There are lots of ways of doing this, and you don't have to do this by any means. It just helps you to get the higher rate of germination. If that's what you're after, this is how it's done. You find a way to get rid of the duds, the little seeds that didn't form properly. And air is a good way to do that. When we clean bean seeds to get the little bits of pods out, I often use a little bit of airflow. Electric fan is good. Just pour the seeds in front of an electric fan and all the little bits of chaff fly away. Well, every now and then a little wrinkled bean also flies away. You should let that happen, because the little wrinkled beans that are smaller and not so filled up, they actually didn't form properly and they're not going to grow. So by letting them go, you wind up with higher quality seeds. You just keep the ones that have the higher germination rate. Here's an example when I was pouring the tomato pulp, and all the pulp, rotten, smelly pulp was pouring off the top, but the seeds had sunk down to the bottom. Well, you might have noticed that there were some seeds up at the top that were going over the edge. I lost some of those seeds, but I didn't worry about that because the floaters are usually the ones that aren't going to grow anyway. You can imagine that they're just sort of hollow. They don't have a fully formed baby plant inside them. They haven't stored up lots of food, so they're less dense, and that means they float, and that means I'm happy for those to float off the top, because then I keep the best seeds and I wind up with a higher germination rate. And when we store the seeds, it's fine to do what almost everybody does. Keep your seeds in a paper bag, paper envelope. Any kind of paper packaging is the right place to put your seeds after they're harvested. The reason is, even after your seeds kind of dry and you touch them and you think, well, these seeds feel dry. They're not. Not for the first few months. the first few months after harvest, seeds still have quite a bit of moisture inside them. And if you put them in a sealed package, and that can be just a plastic bag or a plastic container or a glass jar, then a few weeks later, when you look at them, you'll find they're all moldy because the moisture inside the seed is enough to allow mold to grow. You have to let seeds breathe for at least the first few months. I usually wait until about December. Holiday time is a good time to package up your seeds into containers. Until then, let them breathe, let them dry out, and the nice dry air in December is perfect for drying seeds. Here in Ontario, we have really dry air in December, and let your seeds be exposed to that before you put them into any kind of container. Once they're nice and dry, then you can put them in a jar, and or I use jars like you see in the bottom picture. Any kind of plastic package would work too, but it's true that jars are a better container because they don't let any air through. Plastic allows more water vapor through the plastic, even right through the plastic, than glass does. So if you want to keep your seeds fully dry, and you do, then glass jars are really a very good place. If you keep seeds dry and keep them cool, you can double their lifespan. So if you have just seeds in a paper envelope, they'll be good for a few years. If you plan to plant them within a few years, that's totally fine. And that's how most people do that. They don't keep their seeds for more than a few years. So just room temperature in paper is great, as long as you don't have mice. I have lost some seeds to mice, so that is a consideration. But once you have good dry seeds, keeping them colder than room temperature will double their life. And one example is you could put them in a glass jar and put that in the fridge. Now your fridge is a really wet place. If you don't think that your fridge is all that wet, next time you open your fridge, just sort of feel all the surfaces inside and notice that it does feel a little bit moist on the fingers. Your fridge is much more humid than you might think, and that's a terrible place for seeds. They need to be kept dry. But if they're sealed in a jar, then they can be dry inside the jar and cold, and dry and cold is the best place for seeds. And if you're really serious about keeping your seeds for a long time, 10 years, 20 years, then you can freeze them. They have to be fully dried before they go in a freezer, and I'll show you how we do that in a little bit. But the reason is that when water freezes, it crystallizes. So if there's any water inside the seed, or too much water anyway, when it freezes, it'll form crystals inside the seed, and that damages the seed tissue. It can either lower the germination rate or it can kill the seed completely. So fully dry if you're going to freeze, but then the seeds might last for decades. So I'll show you a little bit about the dry and cold. We keep our seeds dry first. If you can't keep them cold, at least keep them dry. I'd rather have seeds in a dry and warm place than a humid and cold place. Okay, dry first. Then if you can manage cold as well, that's even better. Here are two examples of identical seeds. One of them was put in a humid and warm place, and the other one was kept in a dry and cold place. And on the graph, the line going horizontally shows the time in years, and the line going vertically shows the germination rate. So both of our seeds, they start out close to 100%, but then the seeds kept in a humid, warm place, they lose their germination really fast, so that after only 2 years, none of them will grow. But the seeds kept in a dry and cold place, they last for a long time. They last, the last of them are kind of dying out after 17, 18 years. That's a really long time for a seed. But look at how the gray line kind of falls. It starts off almost flat. So after even 4 years, you still have a really good germination rate. Keeping your seeds in a dry and cold place makes an enormous difference. I would, of course, recommend a dry and warm place over a wet and cold place. So let's say maybe an upstairs closet is better than a cool basement if your basement is humid. Okay, dry and warm upstairs, moist and cold downstairs, go for dry instead. But if you can keep your seeds in a nice sealed glass jar in the cold basement, then that keeps them dry and cold. That's even better. This is kind of how you'll see older seeds responding to the age. Here are a bunch of seeds that start off at year 0. They're all at 100% germination. Over time, like after say 4 or 5 years, you'll start to notice some loss of germination, so down to about 80%. And after a few more years go by, you'll notice more loss of germination. But once it gets down to about 60% like this, about 50%, it falls off really fast after that. So when we have a collection of seeds, we test the germination from time to time. And if it gets down to 80%, we think that's still okay. If it gets down to 70%, we think maybe we'll have to regrow those seeds in a few years. But once it gets down to 60 and 50%, we know it's going to fall off really fast after that. So it's time to regrow those seeds. And so, as I said, glass is a better place than plastic. We're really lucky that the absolute best way to store seeds, once they're really fully dried so they can go into a sealed container, remember, wait till December to put them in a sealed container, but ordinary jam jars, all those pickle jars, all those empty spaghetti sauce jars, those are actually the ideal seed storage container. They're well sealed. The glass is airtight, which plastic is not. And as long as the seal on the lid is nice and intact and makes a good seal, they're perfectly airtight. So whatever air goes into the jar stays in the jar, and whatever water vapor comes along outside doesn't get in. They are easy to find, they're inexpensive, and they come in an array of sizes to fit the size of the seeds that you want to put in them. So we use all sizes of these jars depending on how big we need the jar to be. And that is the basis of our seed storage system. I mentioned before I would show you how we dry seeds for freezing. And when we dry our seeds for freezing, we're going to put them in a freezer for 20 years. We expect that some of our seeds will be in backup storage in a freezer, and they'll still be alive 20 years from now, because they're rare seeds that we can't get anyplace. And as long as they're not available from any seed company, and they're only available from a few sources that may or may not have them next year, we want to make sure that we can always grow those seeds. And if they do disappear from seed catalogs, we will have a backup. So we take it very seriously. You don't need to do this. You can set up your own seed bank in your house, because, as I said, we're using the same technology in our homes as seed banks and seed companies use, just well-sealed containers, kitchen sieves. It's really not rocket science, just a difference in scale. So this is how you would do it. The bin on the left is just an ordinary bin, but on the bottom, you might see sort of a white thing in between the jars. That's silica gel. We buy it from craft stores. It's usually used for drying flowers or for other kinds of crafts. We use it for drying out seeds. Now, silica gel is a substance that it's really like little glass balls that absorb moisture from the air. That's why it's used for drying flowers. So we put that in the bottom of the bin and then put our jars on top with the lids open. Then we stretched a clear plastic garbage bag over top and made sure it was a nice tight fit. And 3 or 4 days later, the silica gel will have dried out all the air inside that bin and dried out all the air inside the jars, and also dried out the seeds nicely too. So if you're really good at this, you can close the lids without taking the garbage bag off. I'm not good enough. I have to kind of reach underneath and close the lids, but that's okay. That works too. And once the lids are closed, it doesn't matter where you put the jars. You can put them in the most humid place anywhere. As long as the jars are well sealed, the air inside the jar will stay dry and the seeds will stay dry. So we keep the jars in boxes in freezers. And even if the power goes out and the you know, all the frost in the freezer melts and the whole thing gets wet, the seeds will stay dry. And at freezer temperature, the seeds should last at least 20 years. And that's how we do it. Now, what about the silica gel? Once it absorbs moisture, it stops working. It doesn't absorb moisture forever. So usually they have a few indicator crystals. They're usually blue, and when they turn from blue to clear, it means that they don't absorb any more water. So you take this, it's just little glass balls, put it on a baking sheet on low heat in an oven, and in no time at all, the little crystals turn blue again, because that dries out the silica gel, and then it can be used again. It'll absorb moisture again. And you can repeat this hundreds and hundreds of times. It's a very durable substance. So once you have some, you can just keep on drying it out and reusing it. So we talked about how to make sure our seeds germinate well. And the whole point of it is that we start out with seeds that are good, that they've been harvested when the plants are fully ripe, when the fruit and the seed heads and the pods are fully ripe. And that means the seeds are well fed, so they have lots of food to eat while they're waiting to germinate. We've dried the seeds properly, being careful to make sure they're fully dry when we put them in a sealed container, and then kept them dry and cold. And under those conditions, they should last a long time. So they germinate when we want them to. Now we'll look at how we get our seeds to grow into the plants that we want them to grow into. So the first thing is how we prevent them from cross-pollinating with other varieties. And the second is how we remove off-types that would change the variety over time. And then we'll talk about how to make sure we have an adequate population of seeds. So there are three kinds of pollination. You've probably seen this before, that some plants self-pollinate, some are pollinated by insects, and some are pollinated by wind. In this example, there are tomatoes that are self-pollinating, squash is cross-pollinating, and corn is wind-pollinated. So we have seeds harvested from tomatoes will tend to be exactly the same as the seeds that were planted to grow that plant. Even if you have different tomatoes nearby, they won't tend to cross, and the reason is every tomato flower pollinates itself. This bee, you'd think, might be carrying pollen from one flower to another, but in fact, on that tomato flower, the bee is hanging upside down on a tomato flower, and that cone that she's holding on to, all the pollination happens inside that cone. The only open part is a tiny hole at the tip of the cone, and what she's doing is she's trying to shake pollen out of that flower like a salt shaker so that she can eat it. Bees eat pollen, it's their food. So she's a bumblebee, and she's collecting pollen by shaking it out of the flower, and she'll eat it, but she can't somehow shake pollen back up. It's really hard to shake salt up into a salt shaker, and so that tends to not happen. If she goes to different tomato flowers while she's feeding, she'll only get pollen out, not put pollen back in. And that's why tomato flowers don't cross with each other. And that means you can save the seeds from those different tomato plants, and they stay the varieties that they were. Same thing roughly with beans and peas. That's a pea flower on the right. It's a very closed up kind of flower, so the same kind of thing happens. Whereas wide open flowers like squash are freely cross-pollinated by insects. This is a squash bee in the middle who will fly from flower to flower and feed on the pollen, but also, since I think it's a he, will be able to pick up pollen on the way. Some of it sticks on his body, sticks on his underside, and so he carries the pollen and drops it off from flower to flower while he's eating. And that means who knows where the pollen came from that fertilized your pumpkin. It might have been from a zucchini plant somewhere, or it might be from an acorn squash somewhere. So your pumpkin seeds might be half pumpkin and half zucchini, half pumpkin, half acorn squash, and when you plant them, they won't necessarily be pumpkin. How far away would you keep different kinds of squash? Well, as far as this bee can fly is the answer. And that could be a few hundred meters at best, and possibly more. So in another session, we might talk about hand pollinating, but you can basically take the pollen from one flower and hand pollinate another flower, and by using clothespins or masking tape, keep the flowers closed so the bees can't get in, and control the pollination that way. If you're curious about that, look it up. It's called hand pollination of squash, and it's not that hard to do. It's worth trying. You'll find instructions out there. Cross-pollination by wind makes it much more difficult. This is spinach over on the left, and corn on the right. It's a picture of corn. These are examples of plants that have pollen that's really lightweight and tends to travel for a great distance. It's very difficult to save seeds reliably from corn if you have any other corn growing in the miles nearby. This might not look like spinach on the left, but indeed that is a female and a male spinach plant. The little green balls on the stem of the female plant, those are the seeds, and they're not ripe yet. They have to be brown and dry before they can be harvested, but they've been pollinated by pollen floating through the air from the gentleman on the right. And once those little green balls turn brown, they can just be stripped off of the plant and either planted right away to get a second crop of spinach, or kept until next year. I haven't purchased spinach seeds in years and years. This is just a really easy way. Let your spinach go to seed, or not all of it. Eat your spinach, but let a few plants go to seed, and you can get your own spinach seed this way. But if we're serious about this, we want to make sure that we're following some standards. So Seeds of Diversity has two standards. We talk about community seed and commercial seed. They refer to standards that are easy for people to manage in a small scale setting. So keeping different tomatoes only 5 meters apart, that will give you good results most of the time, because tomatoes are self-pollinating for the most part. Beans are more self-pollinating. If you keep them 3 meters apart, you should get good results without them crossing. And squash, I said that squash bee could travel for a few hundred meters. 400 meters is probably enough from other squash for there not to be cross-pollination, but you might get a few percent of off types of crosses within your squash at 400 meters. For commercial standards, those aren't good enough. Commercial seed companies have to use greater standards. But this is kind of a comparison of those standards. And for seed banking purposes, we go for the commercial standards. Commercial seed companies also have 2 different grades of seeds. The ones that they produce, called market seeds, that they just produce and sell. And they're not intended to be perfectly pure. They're intended to have a little bit of cross-pollination, but only a little bit, so that the market will accept that. But if you're growing seeds and you're going to use those to grow more seeds, and you're going to use those to grow more seeds, then you have to have a much higher standard of purity. It's called stock seeds, because every little bit of cross-pollination in those seeds will add up generation after generation after generation. So they have their own stock seeds that they're growing to very, very high standards, isolated from other varieties, in order to keep them as pure as possible. And that's, imagine you're a seed bank. Imagine you're collecting heritage seeds that you can't get. And the only way to get those seeds is to grow your own. We have to maintain our very rare seeds at that very high standard, because the only source of seeds is our own seeds. And we don't want them to lose their identity by getting crossed with other varieties. So we have to have those same standards as commercial seed companies do. Here's an important thing. When you grow a bunch of tomatoes, say lettuce, beans, whatever it is, look at all of the plants at all stages, at the seedling stage, at the transplanting stage, at the flowering stage, at the produce stage, and look for any that are not quite the way they should be. This cabbage, it is one of the cabbages. You can sort of see where it came from. it's a flathead cabbage, and all the others are supposed to be roundhead cabbages. I don't know how a flathead cabbage got into my special heritage cabbages here. It's a variety called Houston Evergreen. It's a roundhead cabbage. One of them was a flathead. Shouldn't have been there. So I ate it for dinner. The result of that is that I will not have that characteristic in my seed crop, that the next generation, when I plant the seeds, I shouldn't have introduced those flathead genes into that next generation. I've removed those flathead genes, and I should just have round-headed cabbages. It's a sort of a maintenance thing, but you'll wind up with much better quality seeds over time if you always look to remove anything that isn't what you want to be saving. And population size is a crucial thing that a lot of seed savers haven't maybe heard about, but in seed production circles, it's really well known, and we need to talk about it more in order to do seed saving, even backyard seed saving, better. The thing is that even in a row of lettuce, say, where they all look the same, you'll look at that row of lettuce and say, these lettuces are all identical, but they're not. They've been made to be identical by removing all the off types. If there were a different color, different shape, different size, that would have been removed, just like I said in the previous slide, rogued it out. But you can only rogue out the things that you can see. What about resistance to diseases? Maybe there's a lettuce disease, which you don't have right now, but some of those lettuces have a resistance to it. You'd want to save seeds from those lettuces to make sure you keep that quality. What about drought resistance? You'd see that in a drought year, but you might not see it in an ordinary year. Well, you want to keep seeds from all of those lettuces so that you don't lose that. What about a new disease that hasn't come up yet? Well, we all know that new diseases come along from time to time. That's the big thing we all just learned. Happens with plants too. So maybe there are diseases we don't even know about that will affect lettuce, that some of those lettuces have a natural resistance to. Make sure you save seeds from those to make sure we don't lose it. So you have to save seeds from all of your lettuces to make sure that you capture all the genes. Well, you have to save seeds from enough. And what's enough? That's what we call a population size. For very inbreeding plants like tomatoes, beans, and lettuce, which are self-pollinating and therefore inbreeding, only 10 or 20 plants will be enough to capture all the genes. We don't want people to just collect seeds from one lettuce plant, because you'll lose the drought resistance and the disease resistance and the, all those things that you can't see, but they're in there in other plants that you didn't save the seeds from. But 10 or 20 plants, that's plenty. That should do it. For partially inbreeding plants, it's more like 40, and that's peppers for outcrossing plants, which is most plants that have wide open flowers. So our biennials are good examples of that. A lot of flowers. We're talking about now collecting seeds from 80 plants, and that seems daunting. That seems like a lot. And so we're working on strategies for collecting seeds from different groups of plants that might be grown by different people in different places, and combining those seeds together so that we're not creating bottlenecks in the genetics, but having a more diverse population of seeds that are still rogued to the characteristics that we want. So they're all the right color and the right shape and the right flavor, but they can have diversity in disease resistances and diversity in drought resistances, and things that we can't see. So that's something I encourage you to learn more about. I don't have time to get into it in a lot of detail. It's a really important concept in seed saving for the seed saver who's going to the next level, to understand population sizes and to work on that. And that's what we will help you learn about. But we don't have time to go into that too much more. So here we've covered how to get your seeds to germinate well, and how to make sure that they grow into the plants that you expect. And those are the very same techniques that every seed company uses, and anyone who works for one of those big seed banks that you see in the magazines sometimes, they're doing the same things in the same ways. So you can do this in your backyard, your garage, and your just the same way that it's done by the professionals, just with different scale. That's all. A few other things that people would ask about before I wrap up. Biennial vegetables is an advanced topic. Biennials are plants that produce their seeds in the second year of their life, not in the first year, like most of our garden plants that we're used to. And so biennial vegetables, beets are an example. If you grow beets, you don't see flowers and seeds the first year. I used to pick apart my carrots when I was a kid. This is true. I used to pick apart my carrots looking for the seeds. I thought there should be seeds in there somewhere. And of course, the seeds don't grow in the roots anyway. I've learned a lot since then. Seeds grow in flowers, but we don't see carrot flowers. Actually, we do, because Queen Anne's lace, the common roadside weed, Queen Anne's lace, those are actually carrots. That's exactly what carrots are. They've gone wild. The original wild relative of carrots from Middle Europe is everywhere in Canada now. It's an invasive species technically, but it's also our food. So if you grow carrots for 2 years in a row, you'll get Queen Anne's lace flowers and carrot seeds. And over on the left, that funny looking bunch of long skinny things, those are little pods of kale seeds. Kale also usually doesn't produce a flower and seeds in the first year. It needs 2 years. How would you get carrots and beets to grow in a second year? Well, you have to keep them over winter. And since in Ontario they freeze and cannot survive over the winter, you have to dig them up and store them and plant them back in the second year. So this is sort of something that if gardeners get into seed saving when they want to learn something, seed savers get into biennials when they want to learn something. It's a beyond the basics kind of a thing, but it's not that hard to do. If you have carrots that live in your fridge for several months, then you could probably manage to keep carrots in a fridge and plant them back. The trick is to keep them cold but not freezing, and humid but not moist. So not too wet. See these beets? They're just a little bit humid, but we don't water them because they'll rot. So it's a delicate balance between keeping them a little bit sort of moist but not too moist, and keeping them someplace cold but where they won't freeze. And finally, how often should you regrow your seeds? We do germination tests. There is no rule of thumb on how long your seeds should last or when you should regrow them. The only way to know is to test them and see if they're still going. So you germinate seeds at the correct temperature for them if you like to, or if lettuce seeds like to germinate at a lower temperature and pepper seeds like to germinate at a hotter temperature. Do your germination tests at those temperatures. Look up the optimal germination temperature for your seeds and do your test at that temperature, because that gives you the most meaningful results. And how many seeds make a good test? For a commercial germination test, it's 400. And that's too many for me. I don't even have 400 seeds of some of the varieties I have. But that's so that you can get an exact number. If you've ever had a package of seed that says tested for germination 97%, well, they want to know, is it 97 or 96 or 98? So they test 400 seeds to get it that precise. You don't need that. You need to know, are your seeds still good to grow or not? And 10 or 20 seeds will give you enough of an indication of that, yes or no. You see some good sprouts coming up in your test, then you know the seeds are still good for a while. And if only a few of them are growing or only half of them are growing, then you know it's time to grow more. So just 10 or 20 seeds is plenty for a yes or no test. And so I thank you very much for the chance to talk to you. I will take some questions, but I'd like you to know where to find more information. We have information on our website, seeds.ca, about other Seedy Saturday events and webinars that are just like this one. And we feature on our website a complete list of all the varieties that are offered by Canadian seed catalogs. We don't sell seeds, but we have this as a resource so we can know which varieties are worth saving and which are rare and which aren't. And we offer that to you so you can find anything you're looking for. If it's available in Canada, you'll find it in our index and you'll know where to buy it. And we also have a seed exchange of that 7/8 of the seed diversity that you can't find in seed catalogs. A lot of those are in our member seed exchange of seed savers offering seeds in their backyards to each other. So you can find a lot more information there. And thanks, I'd be happy to answer any questions that have come up.
Oh, thank you very much, Bob. That was amazing. That was a lot of information. So hopefully some good advice for people. We do have some questions coming in and not a lot of time, so I'll try and get through them. The first question from Christina. So when you were showing the jars and you put the silica gel packet in there, she was asking, would filling the empty space in the glass jar with wax paper or paper towels help with preventing moisture accumulation?
It might, if the paper towel itself is drier than the air, then it could help to absorb some of the moisture. I think the problem might be that then the paper towel would have the moisture in it. So when you seal the jar, you're trapping that moisture right in the jar. And it's probably better to get the moisture out of it in the first place.
Vanessa asks, hi Bob, I'm wondering if the tomato seed fermentation method might work for other seeds that are held in the wet jelly-like substance. So she's thinking grapes, cucumbers, strawberries.
Exactly, yes. Tomatoes is the classic example because lots of people save seeds from tomatoes. But if you let a cucumber grow to its fully ripe form, which is usually it turns yellow and gets kind of spongy, you'll find that it has the same kind of viscous jelly around the seeds, and that's the perfect technique for that. And then other seeds that are just hard to remove. I've even done that with eggplant seeds. They're just hard to pick out of the eggplant. So just let the whole thing rot and rinse it off the seeds. That's an easy way to clean the seeds off.
Jen asks, is there a realistic way of hand pollinating 80 or more brassicas, my goodness, or other biennials instead of using introduced insect larvae / pollinators into an isolation tent? So if realistic to hand pollinate these instead of any insects, are there any tips?
Okay, so I have hand pollinated small numbers of cabbage flowers with a paintbrush. You get a really soft camel hair artist's paintbrush so that it's soft enough that it doesn't damage the flower, and you can transfer pollen from one flower to another. It's super time consuming, and you still have to keep the space enclosed so other insects aren't getting in there. But yeah, that is done if you have the time and the patience for it.
Ruth Ann is asking, do you have book suggestions for detailed information about seed saving?
Yeah, you should go to your library and look up books on seed saving. There have been several printed. I would be remiss not to mention the one that Seeds of Diversity sells. You'll see it on our website. It's sort of more complete version of what I've done here. But there are others, of course, and I would recommend one by Suzanne Ashworth, which I'm not sure is still in print. I would recommend one from the Seed Savers Exchange as well, which I think it's called The Seed Garden. It's a big one and very complete.
Mm-hmm. Yes, I have this Seeds of Diversity book, and it is very helpful. It includes all your common vegetables and a few of the ones that maybe you're not growing, so it's a great resource. I have one. So those rare varieties that you're keeping in the freezer for 10, 20 years, as long as possible, are you continually growing them as well to get more seed, or are you just locking them away?
We are not locking them away. One of the criticisms about seed banks is that it's great that all those seeds are saved for the future, but we can't get them now. And so our organization puts accessibility first. We sort of stand in the middle between seed companies and seed banks, and we call what we do a seed library. And the importance of that is that library is about access. Banks are about safety, and so we take the safety part of it seriously, but we use seed banks to back up our seeds, and we focus our effort as much as we can on making sure people can get the seeds, because that has to be part of it.
Perfect. And so you did mention that the tomatoes have to be 5 meters apart, but you also said that tomatoes are self-pollinating. So why still the 5 meters? Why do you have to do that?
Every now and then, there's a flower that that hasn't really formed properly, and it has a bigger opening than the others. So it becomes very difficult to see that later on when you harvest the tomatoes. So just to be on the safe side, and there is some cross-pollination that happens from time to time because it's nature, and nature doesn't follow the rules of the book.
Yes, that is true. It's wasn't it in Jurassic Park? Nature will find a way. Life will find a way.
It does. It always finds a way around our rules of thumb.
Ian would like to know, can we buy any books from Seed Diversity as a PDF?
We don't currently do that. No, I think we're modernizing gradually, but currently only print.
All right. Well, I think we're run out of time here, but it has been wonderful having you here and speak with us about seed saving. This has been a great resource for people. We really appreciate you coming on today. So thank you very much.
Well, thank you. If you have any other questions, you'll find our email address at this website, and feel free to ask questions and I'll answer them.
Thank you. All right, thank you very much for joining us today. And don't forget, we have trivia night tonight. So please join us tonight for a little bit of trivia, plants on the brain. So Bob, we'll just ask you to hang on a second.